I'm Hanna. I work on gene regulation. If we take out the DNA of a single cell in our body and stretch it out into a straight line, it's going to be 2 meters long. Fortunately, in our cells, the DNA is tightly curled and wrapped up so that it fits into one single nucleus. However, not every stretch of DNA is packed in the same way. Some regions are more loosely packed and open, providing access to reader proteins that transcribe the genetic information, which is then translated into proteins, the building blocks of the cell. It is very important for each cell that the right regions of DNA are open and the right regions are closed. What regulates DNA accessibility? This is the job of so-called chromatin remodelers. Chromatin remodelers are protein complexes that can open and close the DNA. People who are born with mutations in chromatin remodelers very often suffer from neurodevelopmental disorders, like autism. One of the most frequently mutated chromatin remodelers is the BAF complex. In general, BAF opens up tightly packed regions of chromatin to make them accessible for reader proteins. However, we have seen that even though BAF generates DNA accessibility, this does not always lead to the reading of the DNA. We assume that BAF has to interact with other proteins so that the DNA is not only accessible, but can also be read. In my thesis, I want to find out which proteins these are. The problem is that we have no idea which protein could have such a function. Therefore, we are going to look at every single gene in the genome. This means 20,000 genes. How can I accomplish this? I'm going to take a pool of cells, and in every cell, I delete a different gene. I can achieve this thanks to the novel Crispr Kiss9 technology, a genetic tool for cutting genes. Since every cell is now missing a different gene, this means every cell is missing a different protein. The cells that I'm using are special. They have been modified. If BAF finds its partner to read DNA, the cell will die. However, if BAF can't find its partner anymore, because the gene encoding for this protein has been deleted, the cell will survive. This means I can just collect the pool of surviving cells, and via next generation sequencing, I can figure out which genes were deleted. In this way, I can find novel interactors of the BAF complex that are necessary to activate DNA reading. With this project, I hope to deepen our understanding of how BAF complex functions, which will in the future help us to comprehend how neurodevelopmental diseases arise and how to effectively treat them.